Hello students, I am Mrs. Ghodake-Patil working at Swami Ramananda Bharati Vidya Mandir and Junior College Tasgaon. Today we study the topic, seventh topic, cell division, and from this topic this topic contains 4 marks compulsory with option 6 marks. So this topic having the different stages of cell division that we are going to study in detail, but see how the cell divides during the process. It shows different stages. Now today we study the mitosis and see that phases of mitosis, that is namely prophase, prometaphase, metaphase, anaphase and telophase. With this diagram, we can understand the cell divides with the cell division, that is mitosis, completes with the different stages. Now we study one by one. First, we study the cell cycle. The cell cycle actually nothing but these are the events occurring during the life of a cell. It consists of two phases. First one is the interphase, second one is the M phase. Interphase is nothing but it is the stage between two successive cell division. Successive पाठोपाठ दोन divisions असतात त्याच्यामधली phase आहे. It is the longest phase during which the cell is highly active. That means during interphasic division the cell becomes active, more active, and prepare itself for the division. It is divided into three phases, and that three subtypes or three phases are nothing but G1, S phase and G2 phase. That G1 phase is growth phase. Then we study one by one, that is the interphase. First one is the G1 phase, or it is also known as the first gap period or first gap phase. So first see the diagram. That diagram contains the different stages or shows different stages during the cell division. That is the G1 phase or that first gap period or first gap phase. Then second S phase, then G2 phase and M phase. That means these are the interphasic divisions and this is the M phase. Thus with the help of these two phases the cell division completes, and that is the cell cycle. The diagrammatic representation shows the cell cycle. So first we study the G1 phase or first gap period, and it begins immediately after cell division, in which RNA synthesis, protein synthesis and synthesis of membranes takes place. Then second, that is the synthetase phase or synthesis phase, or it is simply known as the S phase. So see in the diagram, in this phase DNA synthesized. So that amount of DNA is double, that is replication takes place, and also synthesis of histone proteins takes place. That means the protein named as the histone, the synthesis of that histone is also takes place during the synthesis phase. Then third one, that is the G2 phase, or it is also known as the second growth period or second growth phase. Metabolic activities which are essential for cell division occurs during G2 phase, and also different types of proteins necessary for cell division are synthesized in this phase. In animal cell, a daughter pair of centrioles or daughter cells appear near the pre-existing pair. Then next one, that is the M period or M phase or period of, it is also known as the period of division. M stands for mitosis or meiosis. M phase involves karyokinesis. That karyokinesis is nothing but division of nucleus. And another phase of the M phase, that is the cytokinesis. That means division of cytoplasm. And then we study the types of cell division. Actually, there are different types of cell divisions which are occurs in the animal cells. And these are first one amitosis, then mitosis, and then meiosis. So, see, amitosis, or it is also known as the direct division, which takes place in the unicellular organisms. Then mitosis, or it is also known as the indirect division, and meiosis, or it is also known as the reductional division. We study one by one. Mitosis can be performed by haploid or diploid cells, but meiosis only performed by the diploid cell. Haploid means N, diploid means 2N, and that thus the meiosis only occurs in the diploid cells. Then first study the amitosis. In this stage, the nucleus longest and construction appears along its length. The construction deepens and divides nucleus into two daughter nuclei, and thus two daughter cells are formed. This type of division is observed in unicellular organisms, abnormal cells, old cells, and fetal membrane cells. That means these are only occurs in the unicellular organisms. Then second, that is the mitosis. In this cell divides to form two similar daughter cells which are identical to parents. म्हणजे जी पॅरेंट सेल असेल ज्याचं सेल डिव्हिजन होते मायटोसिस होतं त्या सेलमध्ये that is known as the parent cell. And the cells which are formed during mitosis, these are similar, that is identical to the parents. It is completed in two steps, that is the karyokinesis and cytokinesis. See, these are the stages of mitosis. This is the only diagrammatic representation of mitosis. First interphasic cell, then it completes two stages, that is it continued into two stages, prophase. In prophase there are two stages, early prophase and late prophase. In early prophase, the cell or centrioles or DNA material or chromatin material or nuclear material is in the form of thread-like structure, and after this it becomes like this, that is the condensation of the chromosomes takes place in the prophase. Then this is the metaphase, equatorial division. We study after the diagram or in detail the divisions of mitosis. This is the anaphase and this is the telophase. This is only, म्हणजे हे डायग्रॅमॅटिक रिप्रेझेंटेशन थोडं आपल्याला समजण्यासाठी अगोदर दिलेलं आहे. वन बाय वन आपण स्टडी करणार आहे. सी द डायग्रॅम. दीज आर द टू स्टेजेस ऑफ द प्रोफेज, दॅट इज द अर्ली प्रोफेज and it is the late prophase. Centriole is formed, paired centriole, and the condensation of chromosome C in the late prophase. Then see, these are the diagrams of early prophase and late prophase. In the early prophase, chromosomes or duplicated centrioles are formed and nuclear material shows the thread-like chromosomes. And in late prophase, the nuclear membrane, that means this covering, disappears and condensation of chromosomes takes place. So, see the mitosis. In this cell divides to form two similar same daughter cells which are identical to the parent cell, and it is completed in two steps, that is karyokinesis and cytokinesis. Karyokinesis is again subdivided into prophase, metaphase, anaphase, and telophase. So we study one by one as seen in the diagram, that is the prophase, condensation of chromatin material, migration of centromeres, disappearance of nuclear membrane takes place, and each chromosome is with sister chromatids. See here the pairing of the chromosome attached to the centromere. न्यूक्लिओलस स्टार्ट्स डिसॅपियर. ह्याच्यामध्ये न्यूक्लिओलस जे असतं इथं दिसतंय हे डिसॅपियर व्हायला सुरुवात होते आणि सेंट्रोझोम स्टार्ट्स मूव्हिंग टुवर्ड्स द अपोजिट पोल. रिस्पेक्टिव्ह पोलला म्हणजे ह्या एका टोकाला आणि ह्या एका टोकाला सेंट्रोझोम मूव्ह व्हायला सुरुवात होते किंवा दोन रिस्पेक्टिव्ह टोकांकडं अपोजिट पोल्स म्हणतात त्याला. दिस वन अँड दिस वन. इकडं सेंट्रोझोम्स इथं दाखवलेलं आहे. ह्या पद्धतीनं सेंट्रोझोम दोन अपोजिट पोल्सना मूव्ह होतं. त्याच्यानंतर नेक्स्ट फेज आहे प्रोफेज नंतर, दॅट इज द मेटाफेज. सी इन द डायग्राम अगेन that metaphase. In the metaphase, spindle fibers starts to form. Then here at the equatorial region, the chromosomes appears at the equatorial region in between the poles. That means exact central region of the cells, and that chromosomes are thick, coiled and lying in the center. That is the known as the metaphasic plate. Remember, the spindle fibers attached to the respective poles, and this is formation of metaphasic plate or metaphase plate. So in the metaphase, chromosomes completely condensed and appeared short. Centromeres and sister chromatids becomes prominent here. Then all chromosomes are arranged at the equatorial plane of the cell, and this is called as the metaphase plate. Mitotic spindle is fully formed. Here the spindle fibers are formed, and centromere of each chromosome divides into two, this one and this one, and each being associated with the क्रोमॅटिड्स. अशा पद्धतीनं मेटाफेजमध्ये आपल्याला मेटाफेजिक प्लेट किंवा क्रोमोझोम्स जे आहेत ते इक्वेटोरियल प्लेनला आलेले दिसतात. हे प्रत्येक फेजचं एक वैशिष्ट्य आहे. प्रोफेजमध्ये थ्रेड लाईक अँड देन लेट प्रोफेजमध्ये कंडेन्स्ड फॉर्ममध्ये क्रोमोझोम्स आहेत. मेटाफेजमध्ये ॲट द इक्वेटोरियल प्लेन अँड फॉर्म्स द मेटाफेजिक प्लेट. Then next one, that is the anaphase, third stage of mitosis. See in the diagram, the chromatids moving opposite poles. हे क्रोमोझोम्स जे मेटाफेजमध्ये इक्वेटोरियल रिजनला होते, that moves towards the respective poles. Thus in the anaphase, in this phase, the chromosomes of each or centromeres of each chromosome separate and form two chromosomes called daughter chromosomes. These are the daughter chromosomes that move opposite. The chromosomes which are formed are pulled away in opposite direction by spindle, that are the spindle fibers, which brought these chromosomes to the opposite poles. Anaphase ends when each set of chromosome reach at the opposite pole. ह्या दोन टोकांकडं हे क्रोमोझोम्स जोपर्यंत म्हणजे ह्या एंडकडं जातात त्या स्टेजला ॲनाफेज जी आहे ती संपते. देन द नेक्स्ट स्टेज, दॅट इज द टीलोफेज. मायटॉसिसची लास्ट स्टेज. ह्या टीलोफेजमध्ये क्रोमोझोम्स आर ॲट द पोल्स. क्रोमोझोम्स जे आहेत ते रिस्पेक्टिव्ह पोल्सकडे आलेले आहेत अँड आर बिकमिंग मोर डिफ्यूज स्ट्रक्चर दिसतं. द न्यूक्लिअर एन्व्हलप इज रिफॉर्मिंग. दिस इज द न्यूक्लिअर एन्व्हलप अँड दॅट इज अगेन फॉर्म्ड विच डिसॅपियर्स इन द फर्स्ट स्टेज और लेट प्रोफेजमध्ये. अँड सायटोप्लाझम पण डिवाईड व्हायला सुरुवात होते. ॲक्च्युली टीलोफेज जी आहे फायनल स्टेज म्हणजे न्यूक्लिअर मटेरियल जे आहे ते इथं डिवाईड झालेलं आहे. The chromosomes with their centromeres begins to uncoil at the poles, and the centromere lengthen and lose their individuality. Separation त्याच्यामध्ये दिसत नाही. The nucleolus again reappears in this stage, and the nuclear membrane appears around the chromosomes. Spindle fibers break down and get absorbed. Spindle fibers पूर्ण disappear झालेले that are absorbed in the cytoplasm, and thus the two daughter nuclei, this one and this one, these are formed. Then actually there are some significant points during that division, and that is, see, cytokinesis. That means after the karyokinesis, cytokinesis is formed. See that diagrammatic representation, that is the cleavage furrows formed in the contractile ring form microfilaments, that daughter cells are formed. These are the in animal cell and this is in plant cell. There is difference between the division that is known as the cytokinesis. Vesicles forming the cell plate. सायटोप्लाझम डिवाइड होतं त्यावेळेस सेल प्लेट फॉर्म होते. इथं पॅरेंट सेलची सेल वॉल आहे, इथं सेल प्लेट क्लिअर डिस्टिंक्ट झालेली आहे आणि अशा पद्धतीनं एका सेलच्या नवीन सेल वॉल फॉर्म होते आणि एका सेलच्या दोन सेल्स फॉर्म होतात, दॅट आर नोन ॲज द डॉटर सेल्स. म्हणजे ॲक्च्युली सायटोकायनेसिसमध्ये सायटोप्लाझम डिवाइड होताना ॲक्च्युली द डिव्हिजन ऑफ सायटोप्लाझम इंटू टू डॉटर सेल्स इज कॉल्ड ॲज द cytokinesis. The division starts with the construction, and the construction gradually deepens and joins the center and divides into the two daughter cells. This process of division of cytoplasm is perpendicular to the spindles, and thus mechanism of cytokinesis is the characteristic of animal cell. But in plant cells, the formation of new cell wall begins with the formation of a simple precursor, and that is called as the cell plate, that represents the middle level between the walls of two adjacent cells. Then we study the significance of mitosis. Mitosis is called as the equational division, therefore chromosome number is maintained constant. It ensures equal distribution of the nuclear and cytoplasmic content between the daughter cells, and both, that means qualitatively and quantitatively. Therefore, the process of mitosis also maintains the न्यूक्लिओसायटोप्लाझ्मिक रेशो. न्यूक्लियस आणि सायटोप्लाझम ह्याचा रेशो मेंटेन केला जातो. द DNA इज अल्सो इक्वली डिस्ट्रीब्युटेड. म्हणजे क्रोमॅटिन नेटवर्क किंवा जिनेटिक मटेरियल जे काय आहे क्रोमोझोम्सच्या स्वरूपात, दॅट मीन्स द DNA इज अल्सो इक्वली डिस्ट्रीब्युटेड. इट हेल्प्स इन ग्रोथ अँड डेव्हलपमेंट ऑफ ऑर्गॅनिझम. ऑर्गॅनिझमची वाढ होण्यासाठी आणि ग्रोथ अँड डेव्हलपमेंट होण्यासाठी मायटोसिसची गरज आहे. ओल्ड अँड ओर्न आउट, दॅट मीन्स ओल्ड अँड ओर्न आउट, ज्या मृत झालेल्या आहेत अशा सेल्स रिप्लेस्ड थ्रू मायटोसिस, अँड इट हेल्प्स इन द सेक्शुअल रिप्रोडक्शन ऑफ द ऑर्गॅनिझम अँड व्हेजिटेटिव्ह प्रोपेगेशन इन प्लांट. व्हेजिटेटिव्ह प्रोपॅगेशन प्लांट्समध्ये प्रोसेस होते, रिप्रोडक्शनचाच टाईप आहे तो, आणि ॲनिमल्समध्ये सेक्शुअल रिप्रोडक्शन अशा पद्धतीनं होत असतं. देन नेक्स्ट, दॅट इज द लिटल बिट पॉईंट, दॅट इज द डेथ ऑफ सेल. Actually, death of cell is completed within the two steps, and that are nothing but one is the necrosis and another one is the apoptosis. Actually, necrosis is nothing but it is a form of cell injury which leads to the premature death of cells. Premature death of cells, that is the necrosis. For example, due to scrap or the harmful chemicals, the death of cells occurs, and that is known as the necrosis. Then second type which occurs, that is the death of cell, that is known as apoptosis. It is programmed cell death, or it is also known as the cellular suicide. In apoptosis, cells die in controlled way. Cellular suicide, that is important, and it also helps in eliminating potential cancer cells. In this way, first step of M phase, that is the mitosis, is completed.